So after this tutorial, I would expect you to use this as your reference gauge. This should be your reference about whether you're actually prepared for the practical. I'm not going to go through it now because a lot of the content will make little sense to you. So we'll go back to it at the end. So, just a little bit about the theory. So the context and the definition of cytometry is the quantification of cells or cell characteristics. And the adaptations of cytometry with regards to flow cytometry is the quantification of cells or cell characteristics when they're suspended in a fluid stream using optical lasers and detectors. And the basic concept for the flow cytometer, or the context of the flow cytometer used in this experiment, is to measure the amount of CD4 and CD8 cells present within a patient's blood sample. And the rationale is that if we can quantify CD4 and CD8 cells, we'll get some indication of the degree of which a particular individual may be infected with HIV. And based on the information we get, we can also develop some sort of prognosis for that patient. Okay, so the basic concept is you have a blood sample. It has a whole range of different cells, CD4, CD8, red blood cells, monocytes, eosinophils, cells like this. And what the flow cytometer allows us to do is look, identify and study each one of those individual cells by themselves. Okay, so you have this whole matrix of cells, and the flow cytometer lets us separate these cells and identify them individually. So the measurement of the amount of different immune cell types can be important in disease diagnosis and prognosis. So that's the critical clinical use of the flow cytometer. It lets us quantify different immune cells within the blood, and based on the information you get, it could have importance in monitoring a range of diseases, such as HIV infection or AIDS. So flow cytometer provides a technology for accurate and reliable quantification of a range of different cell types within a patient's sample. The biggest asset of the flow cytometer is it lets us do it in a high throughput manner, which means it can be used in a clinical context in the day-to-day running of the lab. Okay? And you'll see as we move on through the slides, the flow cytometer can actually count millions of cells within a matter of minutes. So, this is the basic outline of a flow cytometer. So here you have the cell sample. In the context of this experiment, this will be a blood sample. So the flow cytometer takes blood up into the machine, and this mixture of blood goes through the nozzle. And as it goes through the nozzle, what happens is it basically creates a laminar flow, which I'll describe in a minute, in which this mixture of cells actually gets streamed into a single cell solution. Now, creating this single file cell solution is critical to the way the flow cytometer works. If the flow cytometer isn't able to create this single file solution, the detector will get mixed signals if two cells are present at the laser at the same time. So, once we create this single cell solution, the cell then makes its way down to the laser. And the laser is shone onto the cell, and the manner in which the cell deflects the light or laser will tell us information on the characteristics of that cell. So the mixture of the blood with the mixture of cells gets put into the flow cytometer. It's streamed into a single cell solution, and this then is exposed to a laser. The cell deflects the laser in a characteristic way, and the laser is deflected into basically the light's deflected and detected by detectors. The detector then transforms that signal into electrical signal, and that's what you see in the computer. Okay, so you need to know the basic principles of a flow cytometer. And the basic principles of flow cytometer start with the fluidics. Okay? And it enables a mixture of cells to be directed to flow in a single file in the fluid stream. And this process of enabling the mixture of cells to go into a single file stream is called hydrodynamic focusing. So when you're discussing or answering this particular question in the practical, you'll get a mark for saying hydrodynamic focusing. Of course, you don't just state hydrodynamic focusing. I would want to say that you understand it. Okay? The next aspect are the optics. And the optics system consists of lasers to eliminate the particles or cells in the sample stream, and the optical filter allows us to direct the resulting light signals to the appropriate detector. This will make more sense as the tutorial goes on. And the last aspect are the electronics. So the electronics transmit the light signal into a measurable electrical impulse, which can be then recorded by the software. Now, I talked about hydrodynamic focusing. I'll also expect you to describe laminar flow. Okay? So blood sample has millions of cells, but the scientist wants to ensure that only one cell arrives at the laser at any one time. And here you can see in the nozzle, you have a whole mixture of cells, but by the time it comes out the end of the nozzle, you have a single cell stream. And the creation of that single cell stream is called hydrodynamic focusing. So, how do we create this laminar flow or hydrodynamic focusing? It's through the use of sheathed fluid. And the sheathed fluid is entered at the same time as the blood sample, but it's entered at a much faster rate. Okay, and here you can see the sheathed fluid entry is actually a lot faster, so it's between 16 to 18 mils per minute, whereas the blood sample is only added at 0.05 mils per minute. And this difference in the rate of delivery creates a difference in pressure between the blood sample and the sheathed fluid. And a difference in this pressure causes the blood sample to basically funnel into a laminar flow, as you can see here. Okay, both diagrams highlight the same thing. The pressure of the sheathed fluid entering forces the blood sample to channel into laminar flow. So what is the sheathed fluid? The sheathed fluid is phosphate buffered saline or HEPS buffered saline. And what this basically is salty water. More or less, it's water with a whole range of salts. And the rationale for that is we want to add a fluid that is isotonic with the cells of the blood. Because what's going to happen if we add water to the blood cells? What happens when you add water to any cell? Osmotic lysis. So it's essential that the sheathed fluid is isotonic with the cells of the blood. The fluidic system consists of a central core through which the sample fluid is injected, and it's enclosed by the outer sheathed fluid. These are pushed through the system at slightly different pressures, and the sheathed fluid is under high pressure and therefore moves faster. As the sheathed fluid moves, it creates a drag effect that causes the sample in the central core to narrow. So when you're describing the fluidics of a flow cytometer, I want you to talk about the difference in pressure between the sheathed fluid and the pressure of the blood sample. And that this difference in pressure causes the blood sample to undergo laminar flow, and this enables the blood cells to be aligned one by one. Okay, so now you've created this single file solution of cells. The next thing the flow cytometer has to do is ensure that one cell reaches the laser point at any one time. Okay? And the point at which the blood cell arrives at the laser is called the interrogation point. Again, that's another mark you'll get for stating that point. And when the laser hits the cell scatters in all directions. And the manner in which it scatters can be described in two ways. One is forward scatter and the other is side scatter. Okay? So the forward scatter essentially is when the light shines on a cell, the cell creates a shadow. Okay, so here you can see the laser or light source going against the cell, and the light that goes above and below the cell creates a shadow of the cell. That's called forward scatter. So forward scatter gives the flow cytometer an indication of the size of the cell. Whereas side scatter involves the light or laser deflecting off the internal organelles of the cell. That'll be the nucleus, the mitochondria, the Golgi. So the side scatter gives us an indication of the internal complexity of the cell itself. So there's 2 points you must know before the practical. Forward scatter tells me the size of the cell, and side scatter tells me the internal complexity of the cell. And at the end of this process, you have a forward scatter detector and a side scatter detector. And this slide basically gives you an indication of how forward scatter works. So if a large cell goes through the laser, you get a wide forward scatter. Whereas if a small cell goes through, you get a narrow forward scatter. Now, starting with a forward scatter detector. This is basically what happens. If you look at this first graph, when the cell is not in line with the laser, you get low or no voltage. But as the cell makes its way and interacts with the laser, the voltage goes up in the detector. And when the cell passes through the laser, the voltage goes back down. And that's why you get this characteristic curve for forward scatter. So, how does that give us information about the size of cell? It's all relative. So, for a small cell like lymphocytes, you'll get a small curve. For a large cell, you'll get a large curve. Now, for side scatter. As I said previously, side scatter gives us an indication of the internal complexity of the cell. Now, if you look at the lymphocyte, there's very limited internal complexity, which is why you get this low voltage. Whereas you move to a monocyte, there's a slightly more complex, and the cell is slightly more complex internally, which is why you get a bigger slope. And of course, neutrophils have a whole range of granules, and added to the fact that they have multi-lobe nuclei, gives us an indication that the internal complexity of that cell is actually quite large, which is why you get the large voltage for the side scatter. So here we have a plot of side scatter here and forward scatter this way. And just to highlight that each individual speckle or spot that you see in that graph equates to one cell. That's a measurement for one cell. So based on this plot, you should appreciate that there's actually, this flow cytometer has measured probably millions of cells based on the amount of speckles or spots that are present. So, If I show you that the subgroup of cells identified as C, the forward scatter is low and the side scatter is low. What type of cell do you think that might be? It's a lymphocyte. Because it's a small cell, therefore the forward scatter is low, and it's got low complexity within the cell, which is why the side scatter is low. If I ask you to identify A, what you're looking at is, I go to the region of A, I go down to forward scatter, and it indicates that it's quite high. So it's a large cell. And I go across to the side scatter, and it's also high. So it's a large cell, and it has a great degree of complexity within the cell. What cell do you think it is? It's a neutrophil. So that's the basic premise of forward scatter and side scatter. Now, as you can see, there are millions of cells within the plot. So there's a whole range of different ways you can analyze this data. And the first one I'm going to deal with is threshold. And threshold is a basic principle of the flow cytometer that lets the user get rid of any unwanted events or broken cells that we don't want to measure. Okay, so say for instance, red blood cells. When you're measuring CD4 and CD8 cells, you have no interest in measuring red blood cells. Threshold gives us the option not to measure red blood cells, but to focus on the leukocytes or the lymphocytes. So the concept of threshold is when the detector is instructed to only detect events that are above a particular threshold. And just to note, the threshold, generally speaking, not all the time, but generally speaking, is determined by forward scatter. And this enables the user to remove noise or debris, so cell debris, or maybe some cell subtypes that you have no interest in. But when you're adjusting the threshold, you have to be careful not to set it too high. If you set the threshold too high, you may actually miss some of the events or cells that you're actually interested in. And this is an example of how a user would set their threshold. Now here you can see this is forward scatter and side scatter. And here you can see a whole group of events that have small side scatter and small forward scatter. Now what the user deduces from that is it's most likely to be debris because they're very small in size and there's very little complexity about them. So what it does is it sets a threshold of 200. Okay, so it moves to 200 and says anything below this we're not going to measure. And this is what happens here. You can see the white line where he set the threshold. So then that wasn't high enough. So he sets the threshold at 5,000. And at 5,000 it's still not enough. You can still see some debris. And in the last graph you can see this individual has set the threshold at 40,000. Do you think 40,000 looks a good fit? Yeah, because as you can see, there's very little debris, which means the data that you get in the file will be the cell subset that you actually want to investigate. So there'll be a 5-mark question on threshold. You'll need to be able to convey what it is and why it's used. So I'm just going to play a quick video to recap flow cytometer.
In this animation, we will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell,
Okay, folks, so that's forward scatter and side scatter. So forward scatter versus side scatter can only, the problem we have now is it can only provide limited information. So it tells us the size of the cell, and it tells us how complex the inside of the cell is. But there are a whole range of different cells present within your blood that are of interest when monitoring disease. And this is where we come up against a problem. Because say for instance we have CD4 cells, CD8 cells, CD4 T helper 1 cells, CD4 TH2 cells, and CD4 TH17 cells. All of those cells have been implicated in a whole range of diseases. But as you can see from the pictures for each cell, they look identical, both in size and in terms of the complexity of the cell itself. And that is why side scatter and forward scatter have limited potential for monitoring cells. And how we get around that is basically to identify characteristics of these cells that make them unique. So what we do is we want to identify receptors that are only expressed in CD8 cells. We want to identify receptors that are only expressed in CD4 cells. So that's the challenge that we have. I'm going to take you through how this process is undertaken. So we did the ELISA last week. One thing you should have took from the ELISA is that we have the potential to create antibodies that can bind to specific proteins. Okay? This concept is also used in flow cytometry. So for instance, if I want to identify leukocytes, the one receptor that is highly expressed in all leukocytes is CD45. So in order to identify CD45 or to identify all leukocytes, what we need to do is to develop an antibody that will bind to CD45. But on top of that, we need to add a fluorochrome to the antibody. Okay? So, we start off by identifying leukocytes by developing an antibody against CD45. Once we've identified the leukocytes, the next thing we want to do is identify the lymphocytes. And the receptor that's uniquely expressed in lymphocytes, not neutrophils, not eosinophils, just lymphocytes, is CD3. So you start off by selecting all leukocytes through CD45. Then within those, you want to identify which of those cells are lymphocytes. CD3 enables us to identify lymphocytes alone. So now we've identified all the lymphocytes, we want to work out what lymphocytes are CD4 and what lymphocytes are CD8. And that's why we then look at CD4 and CD8 receptors. So, the selection criteria. So the user decides they want to, on the first hand, identify leukocytes. So, they create an antibody directed against CD45. On top of that, they want to identify which of these are lymphocytes. So they use CD3. Sorry, just to clarify, CD4 is for leukocytes. CD3 is for lymphocytes. And CD4 and CD8 are to identify CD4 and CD8 cells. So, the problem you have is, if you add antibodies to bind the CD45, you need a way to identify it. And the way we identify it is by adding a little fluorescent tag to the antibody. And these fluorescent tags are called fluorochromes. I'm going to take you through the principle of this process. So here we have a T lymphocyte and a neutrophil. Okay, and they're both present in the same cell, they're both present in the same sample. And you want to identify what's a lymphocyte and what's a neutrophil. So the first thing you do is you add an antibody directed against CD45. So at this point we know we've identified a leukocyte. The next step is we want to differentiate between a lymphocyte and a neutrophil. So we add an antibody that will bind to CD3. Now you can see the lymphocyte now has antibodies bound to its CD3, but the neutrophil has no CD3. So there is no signal for the neutrophil. On top of that, if we wanted to identify the neutrophil, we'll add an antibody against GR1. GR1 is a receptor uniquely expressed on neutrophils. So if we see this colour being identified from the flow cytometric analysis, we know we've identified a neutrophil. The next step is, we know we've identified a lymphocyte, but what type of lymphocyte? So the next step is to add CD8 antibody. And at that point, we now have three colours. We have a green colour for CD45, we have a red colour for CD3, and we have a yellow colour for CD8. And that's the basic principle of flow cytometry. You can add antibodies with different colours to target different receptors. And the more colours you can develop, the more targets you can identify. So what you need to grasp is the importance of creating antibodies directed against unique receptors. And if you're going to target multiple receptors, you have to make sure the colour used for each target is different. If you use the same colour or same colour fluorochrome, the flow cytometer will not be able to differentiate between the different markers. Now, we're moving on to the concept of gating. And gating is the principle where you identify groups of cells step by step. Okay? So process of selecting cell populations of interest and eliminating cells that you're not interested in. So what we have here in the first step is a stain for CD45. What is CD45 stain? Leukocytes. Perfect. So the user's first target is, I want to see what leukocytes are present in this sample. So what he does is he has here, x-axis, him or her, has side scatter and a stain for CD45. Okay? So what they want to identify are all cells that are stained with CD45. So they go across and they identify a group of cells that have high CD45 and they gate them. So this selection box has now selected any cell that has high CD45 and eliminates any cell that does not have high CD45. It takes these cells and moves them on to a different plot. So all the cells you see here are actually from inside that box. And at this point what you have is GR1, which stands for neutrophils, up this side, and CD3 along the bottom. Because we're interested in lymphocytes, we're looking for cells that have high CD3 and low GR1. Because if it has high GR1, there's a chance that this might be neutrophils. So here what the user has done is looked at a group of cells, seen that they are high CD3 and relatively low GR1. And they've gated this group of cells here. They then move on, and all the cells you see in this plot have been taken from this gate. They then move it to another level. They know they have lymphocytes, but they want to work out, are the lymphocytes CD4 or CD8? So what they do, because they're interested in CD8, they go along CD8, look for cells that express high CD8 here and low CD4. And at that point, they know they have CD8 T lymphocytes. Whereas a cell with low CD8 and high CD4 can be identified as CD4 T cells. And that's the basic principle. And what enabled this user to do this was the use of antibodies directed against receptors with fluorochromes attached. And with each target or each receptor that they were interested in had a fluorochrome of different color. So that's the concept of gating. Again, you'll be expected to convey your understanding of the process of gating. So, lasers and fluorochromes. Lasers and fluorochromes are what dictate the amount of targets we can look at. Okay? The lasers, there are a whole range of different lasers you can use, and equally there's a whole range of fluorochromes you can use. When the fluorochromes are stimulated by laser, it causes the fluorochrome to emit a light. And when it emits a light at a particular wavelength, giving this color. The use of different fluorochromes emitting different colors enables us to measure different cell markers. So, here you see a blue laser, and what you have on the right are the range of different fluorochromes or colors that you can use with this particular laser. So there's 1, 2, 3, 4, 5, 6 different colors you can use for the blue laser. Where with the red laser, you have 3 different colors you can use. And with the UV laser, you have 2 different. And violet, 2 different. So the amount of lasers present in a flow cytometer, the more lasers you have, the more options you have in terms of fluorochromes. Okay, and here's another example of different lasers giving different colors or different fluorochrome options. So the amount of fluorochromes that you can use in a flow cytometer is increasing all the time. I think the last time last amount I heard was that you could use a flow cytometer could use up to 18 different fluorochromes because of the number of different lasers that the machine actually had. And here's an example of a laser that emits light at 488 nanometers. Now, if I have 3 different targets that I'm interested in, I'm likely to pick Alexa here, probably BD Horizon here, and PECY7 here. And the reason or the rationale for making that choice is none of the curves interfere with each other. Whereas, for instance, PECY5 here spills in or interferes with PERCP. So when you're going to pick your fluorochromes, you want to make the colors as different and separate as possible. If you pick 2 colors that are similar, there's a chance the flow cytometer won't be able to distinguish between the 2 targets. This is another laser of 405 nanometers, and you can see there are a range of different fluorochromes. Again, if I was to pick if I had 3 markers, I would pick a fluorochrome here, a fluorochrome here, and a fluorochrome here, because there's a nice distance between the different curves. So, we have more lasers, we have more fluorochromes, we therefore need more filters to separate the different fluorochromes or light of different wavelength. And this is basically how the flow cytometer does it. It does it through the use of dichroic filters. And what I want you to be able to convey in the answer for the experiment or the practical is you'll be able to tell me what a long pass filter is. You'll be able to tell me what a short pass filter is. And you'll be able to tell me what a band pass filter is. So a long pass filter, say you have a long pass filter of 560. What that means that any light that's above 560 nanometers will be allowed to pass through the filter. That's a long pass filter. A short pass filter, say of 550 or 560, will allow light that is below 560 to go through. So long pass allows anything above that wavelength through, and a short pass allows any wavelength below that set limit. And the band pass filter is uniquely different because it actually selects a range of wavelength. Okay? So a band pass filter may be 530 to 525. It'll be between 2 different wavelengths. So again, you should be able to convey that. So the use of these different filters lets the emitted light be separated based on wavelength. And this ability to separate based on wavelength lets you separate the light from a red fluorochrome, from a yellow fluorochrome, from a blue fluorochrome. That's how you separate the signals. Okay, so that's an overview of the system. Once the detector detects the signal, it then gets transmitted into electronic signal. And once it's transformed into electronic signal, this is how it's expressed on a graph like this. Okay? So what you see there is the electrical signal transferred onto a plot. And as I said, each speckle or spot in that graph equates to one cell. Okay. Now, what we're going to talk about now is compensation. Because inevitably, if you looked at the fluorochrome graphs, you do get spillover. You do get some fluorochromes interfering with other fluorochromes. There's a concept called compensation. Compensation allows you to use two fluorochromes that are close in color without the machine mistaking one fluorochrome from the other. And I'll take you through that process now. So sometimes small amounts of light from one fluorochrome can actually be picked up by two detectors. The right detector and the wrong detector. So this concept is called spectral overflow. Spectral overflow is when the light for a particular fluorochrome is detected in the wrong detector. And the software, using this concept of compensation, you can instruct the software to use a mathematical calculation to compensate for this. So here we have two fluorochromes, fluorochrome A and fluorochrome B. Now FL1, the detector FL1, has been designated to detect fluorochrome A. FL2 has been designated to detect fluorochrome B. Now as you can see, some of the light emitted by fluorochrome B here is detected in FL1. What that means is some of the light emitted by fluorochrome B is detected by the detector that's designated for fluorochrome A. Which means that the computer will think that there's more of fluorochrome A than there actually is. It's called a false positive. So you can instruct the machine to deduct this portion of the signal. Because we know this portion of the signal isn't fluorochrome A, it's actually fluorochrome B. Okay? And likewise, fluorochrome B or fluorochrome A is only meant to be detected by FL1. But you can see here that some of the light emitted by fluorochrome A is getting detected by FL2, which means this detector will get a higher signal for fluorochrome B than there actually is. So again, you can tell the computer to take away this percentage of the signal, because we know this percentage of the signal is actually fluorochrome A. My advice, folks, is to sit down with the notes and try and get it into your head, because this is a 5-mark question in the practical. You should be able to express the concept that compensation is the ability to account for spectral overlap. And then explain the concept that spectral overlap is when some of the fluorescence for a fluorochrome is detected by the wrong detector. And that based on this spectral overflow, you can create or develop a mathematical equation to adjust for the spectral overflow, which will avoid you picking up false positives. Okay, so that's a 5-mark question. So compensation is required for a flow cytometric experiment because of the physics of fluorescence. And basically because some of the fluorochromes or colors will be too similar in nature, and therefore you need a way to tell the machine how to adjust for their similarities. Using the different fluorochromes to measure different cell markers, enabling different cell types to be identified. Each fluorochrome can be excited at 488. However, the emission detection of each fluorochrome is at different wavelengths. The different emission detection wavelength enables different fluorochromes to be used at the same time. However, the emission detection of one fluorochrome may spill over into the detection wavelength of another fluorochrome, giving you false positives. A mathematical correction can be applied to the data to address this spillover. This is known as compensation. Okay, and there's a link you can follow in your own time to give you more information on compensation. Compensation is probably the most difficult aspect of this particular practical to get to grips with. Okay, so 20 marks of this practical will be based on you working out how much volume the flow cytometer used. Because this is the problem with the flow cytometer. You take your blood, you add your antibody, and you put it onto the flow cytometer. What then happens is the flow cytometer takes up the blood, but we have no idea what volume of blood the flow cytometer used for the analysis. Now, why do we need to know about the volume of blood? Because to diagnose or to develop a prognosis, you need to know how much CD4 cells or CD8 cells are present per microliter of blood. Okay, so it's not just about the number of cells present that the flow cytometer detects. You need to know how many CD4 or CD8 cells are present per microliter of blood, and from that you can then go to the guidelines and develop a prognosis, or in some cases a diagnosis. So the way we do this basically is this. We create fluorescent beads that have extremely high fluorescence. So in this case, the user develops fluorescent beads that are extremely high in PE or blue color and extremely high in the red color, which means they show up on the plot right up in the corner by themselves. So these are the fluorescent beads. And this is the basic concept. If you put 1 million of these beads into 1 ml of blood, and the flow cytometry analysis says that there are 500,000 fluorescent beads present, what volume was used? So you have 1 ml of blood, it has 1 million fluorescent beads. The purple indicated on the graph, they are the fluorescent beads. It's telling me there were 500,000 taken up by the flow cytometer. What volume of blood did the flow cytometer use? Exactly, 0.5 ml, 0.5. So you get the concept. If you know how many beads were in the blood to start with, and you find out how much the flow cytometer took up, you can work out the volume of blood that was used for the analysis. And that's one of the elements to the practical that you'll be expected to do. So, as I said, the blood sample will be spiked with 1 million beads per ml of blood. And then the flow cytometer measure 500,000, therefore you can deduct that 0.5 ml was used. Okay, the next aspect which you can fit into one of your answers is the fluorescent activated cell sorter. This is a really neat aspect of the flow cytometer, particularly when it comes to research. If you want to isolate, for instance, stem cells from a blood sample, there is a way that the flow cytometer can allow you to do this. What you would do is add a fluorescently labeled antibody directed against a cell marker that's unique to a stem cell. When you add this fluorescently labeled antibody and you identify the stem cell using flow cytometer, the flow cytometer has the ability to separate that cell into a different collection container. And what that means is that in research, you can take a blood sample, isolate the stem cells, and then investigate the stem cells. Whereas generally what happens in flow cytometric analysis is you put the blood sample into the flow cytometer, you measure all the events, and then the blood goes into a disposal chamber. The difference with the FACS, or the fluorescence activated cell sorter, is there's a different container for the cells that you're interested in. So it lets you identify cells, isolate those cells by themselves, and subsequently that's when you can do some interesting research with it. So how does it do that? This is basically what happens. The flow cytometer identifies the fluorescently labeled stem cell, and the minute or the second that it identifies as a stem cell, it applies a charge to that cell. In applying a charge to that cell, when it goes on through the flow cytometer, the charge enables it to be separated based on charge. So here you see all the rest of the blood cells going into the disposal, whereas this group of cells we're interested in, because they got an electrical charge applied, it's separated. And that's, again, if you're interested in research, this is where the flow cytometer comes into itself. Another application could be that, for instance, you're interested in a particular leukemic cell, and this will let you isolate the leukemic cell and investigate it to help in the development of a prognosis. Now, going back to the first slide, before you go into the practical, you need to be able to answer these questions. If you can't answer these questions, then you know for sure you're not going to get the marks. Because again, folks, next Tuesday, for all intents and purposes, is almost like an exam. You'll be expected to convey all this information in the workbook that you're provided with. Okay? So that's the main context. The only thing I've left to go through now is to give you a background into HIV and AIDS. So, CD4 helper cells are critical to any adaptive immune response. We'll cover it on Monday. You have two adaptive immune responses possible. Two main ones. One is an antibody response. The other is a CD8 cell response. The importance of CD4 helper cells cannot be highlighted enough, because the B cell response, which produces antibodies, and the CD8 response, which is the CD8 response, both require CD4 helper cells. Without CD4 helper cells, you won't get a B cell response, or certainly you won't get an effective B cell response. And likewise, if you want a CD8 cell response, if you don't have CD4 cells, you're not going to get an effective response. And that's why HIV and AIDS is so devastating. Because HIV and AIDS basically target your CD4 cells. And in targeting your CD4 cells, it basically means that over time, your adaptive immune response becomes depleted. So, you will be asked a question about how HIV infects CD4 cells. So you should be able to tell me that the HIV virus targets the CCR5 receptor that's expressed in CD4 cells. It's also expressed in macrophages, but it's expressed more highly on CD4 cells. So this is the reason why or how HIV targets mainly only CD4 cells, because of this receptor CCR5. So the HIV virus attaches, injects its genetic material, uses the host cell replication machinery to basically replicate its genetic material. And after a series of replications, it more or less causes the CD4 cell to lyse. And when the CD4 cell lyses, you've got a double whammy. One is your CD4 cell's dead. And the second is multiple HIV virions are now released to infect other CD4 cells. And the CD4 cells are critical to monitoring HIV infection. Because when you measure the absolute number of CD4 cells, you can get an indication of how far gone the infected person is. And based on the number of CD4 cells that are present, it can provide information to help you with the prognosis for that particular patient. And the ratio of CD4 and CD8 is critical in the prognosis for HIV infected patients. And as I identified earlier, the ability to quantify the amount of CD4 or CD8 cells is executed or is undertaken using flow cytometry. One thing that I want to convey, or that I want you to definitely take away from this, is quantification of CD4 cells is not the critical factor in diagnosing HIV infection. Quantification of the HIV antigens are what are used to diagnose HIV infection. So folks, when you're answering your questions, keep that in mind, okay? Keep in mind that when you get your CD4 or CD8 data, you actually get more information on the prognosis rather than the diagnosis. The diagnosis is confirmed by measuring markers, physical markers of the virus. Okay? And another thing to take into account is some of the factors that actually, some of the targets for therapy are based on targets that the virus used to replicate. Because if you target the machinery that the virus has to replicate, then it can't replicate, and by default you can reduce the spread. Now, CD4, CD8 cell counts. As I said, flow cytometry is important in monitoring declined CD4 cells. A normal CD4 cell count is from 500 to 1500 cells per cubic millimeter. A normal CD4 to CD8 ratio is around 2 to 1. And in AIDS, this can actually change from 1 to 2. Loss of CD4 cells results in loss of HIV-specific immune response. Basically, what happens is when these individuals start to lose their CD4 cells, they no longer, or they have a very poor adaptive response. And because they have a very poor adaptive response, the bugs that we're exposed to on a day-to-day basis, normal people are able to fend it off. Normal people are able to stop their spread and infection immediately. People with HIV with low CD4 are in a position where those bugs that we were normally able to evade now become a problem. We now get systemic infection. And that's the problem with HIV. Your immune system becomes depleted and you can no longer defend yourself against common pathogens. And also, because the immune system is important in monitoring cancer, you also have an increased risk of cancer. And that's the basic concept of HIV infection. Okay, folks, that's us for the day. If you have any questions, just let me know.